Good morning, all. So, today's session is about Malphigian tubules and its anatomy and histology. Also, we are looking at the hemipteran, coleopteran, and lepidopteran types of Malphigian tubules. So, let's begin. Firstly, Malphigian tubules are tubular organs in insects that are responsible for urine formation. Its anatomy is Malphigian tubules are long, thread-like structure that are located in the body cavity between the midgut and hindgut. They are usually bathed in hemolymph and are close to fat body tissue. The number of tubules varies between species, but most occur in multiples of 2. Histology: the Malphigian tubules are made up of a single layer of cells with a pronounced apicobasal polarity. The apical membrane is covered with microvilli and the basal membrane has convoluted infoldings. Okay, then the function. Malphigian tubules perform many functions including excretion. Malphigian tubules are the site of urine formation in most insect detoxification. Malphigian tubules respond to the changes of insect's environment and induce detoxification genes. Adhesive secretion. Some insects use Malphigian tubules to produce a sticky material for various purposes, such as creating moutique or adhering to vegetation. Bioluminescence. Malphigian tubules can produce bioluminescence. Silk fibers like fiber secretion. Malphigian tubules can secrete a silk-like fiber for building cocoons. Malphigian tubules are used in ecotoxicological studies as models for human renal diseases. So, even the importance of Malphigian tubules are shown here. The Malphigian tubules is a system of excretory and osmoregulatory system found in some insects like myriapods, arachnids and tardigrades. It has also been described in some crustacean species and likely the same organ as the posterior CK which has been described in crustacean. The Malphigian tubules are long, flexuous, thread-like structures about 192 to 312 in number, lying somewhat free in the body cavity and bathed in the body fluid, which is the hemolymph. They are discovered into cephalic group extending as forward as the gastric CK and the caudal group extending as far back as the rectum. So the anatomy. The location of Malphigian tubules are typically located in the abdomen of arthropods. In insect, they are attached to the junction between midgut and hindgut and extended to the body cavity. Also, we looked previously how extended and far back these Malphigian tubules can become in different species. Okay, then numbers and structure. In insect, the number of Malphigian tubules varies, ranging from a few to several dozen depending on the species. Each tubule is a long, slender, and often highly coiled structure that can have various lengths. Connection to digestive system. A Malphigian tubules open into the digestive tract, particularly into the hindgut, where the waste products are processed and excreted. Histology. Epithelium. The inner lining of Malphigian tubules consists of a single layer of epithelial cells. These cells have microvilli on their apical surface, increasing the surface area for absorption and secretion. The epithelial cells are responsible for the active transport of ions, water and waste products like uric acid into the tubules, which are then excreted. Okay, microvilli. The epithelial cells have microvilli on their apical surface, increasing the surface area for absorption and secretion. Then basal lamina. A thin basal lamina separates the epithelial cell from the surrounding hemolymph, which is the insect blood. Also, hemolymph bathing. The Malphigian tubules are bathed in hemolymph, which brings waste products and ion to be excreted. Okay, then we are also again looking at the functions just point by point. Excretion, removing waste products such as uric acid, urea, ions from the hemolymph. Osmoregulation, regulating insects' water in balance by controlling the amount of water reabsorbed or secreted. Ion regulation, maintain the insects' ion balance by controlling the transport ions across the epithelial cells. Malphigian tubules are this is the thing we already discussed, which is found in the posterior regions of arthropods alimentary canal. Each tubule consists of a single layer of cells that is closed off at the distal end with the proximal joining the alimentary canal of the junction between the midgut and hindgut. Most tubules are highly convoluted. Also the tubule number varies. Some insects or most insects are contain accessory musculature associated with the tubules, which may function to mix the contents with of the tubules or to expose the tubules to more hemolymph. The insect orders Dermaptera and Thysanoptera do not possess these muscles, and Columbola and Hemiptera like Aphididae completely lack a Malphigian tubule system. General mode of action. So general mode of action is basically excretion. So let's look at how that works. So preurine is formed in the tubules when nitrogenous waste and electrolytes are transported through the tubule walls. Waste such as urea, amino acid are thought to diffuse through the walls, while ions such as sodium and potassium are transported by an active pump mechanisms. Water follows thereafter. Pre-urine along with digestive food merge in the hindgut. At this time, uric acid precipitate out and sodium and potassium ions are actively absorbed by the rectum along with the water via osmosis. Uric acid is left to mix with feces, which are then excreted. Okay, and here is the alternative modes of action. Complex cycling systems of Malphigian tubules have been described in other insect order. Hemipteran insects use tubules that permit movement of solutes into distal portion of the tubules, while reabsorption of water and essential ion directly to the hemolymph occurs the proximal portion of the rectum. Also, both coleoptera and lepidoptera use a cryptonephrididal arrangement where the distal end of the tubules are embedded in fat tissue surrounding the rectum. Such an arrangement may serve to increase the efficiency of solute processing in the Malphigian tubules. So we see how the alternative modes of Malphigian tubules. So let's lastly look at the types of Malphigian tubules, which are varies or function and morphology differs based on their diet, habitat and metabolic needs. Malphigian tubules in different insect order, such as we are looking here: Hemiptera, Coleoptera and Lepidoptera. So hemipteran Malphigian tubules, which consist of true bugs, cicadas and aphids. Numbers usually 4 to 6 tubules, significantly fewer than in other insect tubules. Structure short, often straight, sometimes forming loops or branching. Functions primarily involved in nitrogenous waste material removal, but also crucial for osmoregulation due to the high water content in their diet. Example: sap-sucking hemiptera like aphids. Some hemiptera, example Cicadidae, have cryptonephrididal systems where tubules associate with rectum to aid water conservation. Adaptation. Xylem feeding hemipterans, example cicadas, need a efficient water excretion due to the high liquid intake in their diet. Phloem feeding species like aphids must balance osmoregulation by removing excess sugar while retaining essential nutrients. Okay, then the next type is coleopteran Malphigian tubules, which mostly consist of the beetle groups. Numbers can range to 2 to 200 depending on the species. Structure is often long, coiled and may show regional specialization. Functions: excretion of uric acid, a water conserving mechanism suited for beetle, especially those are in arid environments. Some species, example Tenebrion species, have cryptonephrididal systems for extreme water retention. These are mostly seen in deserts. Specialized tubule in certain beetle contribute to the production of defensive secretion. Example: bombardier beetles use excretory products in chemical defense. So the adaptations are: desert beetles, example Tenebrionidae, as we spoke before, have modification for water conservation, for extreme water conservation. Aquatic beetles, example Dicticidae, show different osmotic regulation mechanisms to balance salt and water uptake. So we are seeing here how each beetles are specialized in their Malphigian tubules to adapt to its surrounding and to use it to their advantage. Also, the last type is lepidopteran Malphigian tubules. Uh, Consist of butterflies and moths. Numbers typically 4 to 6, similar to hemiptera. Structure tubules are often long and thin, sometimes exhibiting two functionally distinct regions, proximal and distal. We already talked about proximal and distal region. In some larvae, tubules produce silk-like proteins assisting in cocoon formation. Function involved in excretion, nitrogen metabolism, and iron balance. Some lepidoptera larvae excrete high amounts of potassium to regulate osmotic balance. Okay, the adaptations. Larval stages caterpillars often need efficient waste removal due to the high metabolic activity. As we know, larvae are mostly voracious feeders. Adults focus on more water conservation due to the reduced feeding and the reliance on nectar. Most adults don't feed that much and they only rely on nectar, so that's why this Malphigian tubules in adults are focused more on water. Okay, then each group's Malphigian tubules has evolved specific adaptation based on dietary and environmental factors, optimizing water retention, waste excretion, and survival strategies. Although primarily involved in excretion and osmoregulation, Malphigian tubules have been modified in some insects to serve accessory function. Larvae of all species in genus Arcanocamba use modified and swollen Malphigian tubules to produce a blue-green light, attracting prey towards mucus-coated traplines, which we pointed out in the first part of the slide, where we said insects can, Malphigian tubules in some insects can produce bioluminescence. This is that example. Also, in insects which feed on plant material containing noxious allochemicals, Malphigian tubules, allochemicals. Malphigian tubules can also serve rapidly excrete such components from the hemolymph. So it can help the insect or save its life. Okay, these are some references. Also, mostly this slide consists of various information from the internet, from AI to many other sites, Wikipedia, etc., which I am not included in the slide because it's very easily available in the internet. So, thank you.